So we're going to get started with a brief history of molecular genetics. And what we'll do is we'll cover everything that happened between Mendel until modern days of molecular genetics and our understanding of how genes are expressed, okay? So we left it at Mendel developed the theory of particulate inheritance against hundreds of years of this thinking that inheritance happened through blending of characteristics and traits. There was some back and forth between these two camps, and at some point, people came up with a modern synthesis in the early 1900s, saying, well, they can both be true. You could have inheritance happen through particles that are inherited by progeny from progenitors, and you could still have these observed phenomenon of blended characteristics, because what happens is when a given trait is not determined by a single particle but by many, there are mathematical models that can show how you can have a very wide distribution of traits that follow those smooth bell-shaped curves that we see for so many characters like pigmentation, size, and so forth, right? So great, everybody's now more comfortable, everybody's at peace with the idea that heredity is based on particles. Cool. Then the question became, what are those particles, right? So in the, again, early 1900s, biochemists had already isolated from all kinds of living organisms families of molecules, right? That they grouped, the way that we group them today, into these large classes, nucleic acids, among which there was DNA, proteins, polysaccharides or sugars, and lipids or fats. So people knew about those types of molecules. They knew that they had different biochemical properties and whatnot. The question became, which one of these, or which ones of these, are the ones that carry genetic information, right? So we knew those were the building blocks of life. Which of these might be able to be those particles that transmitted information from one generation to the next? So in 1929, a guy named Griffith was working with these two strains of a bacterium called Streptococcus pneumoniae. And what was particular about these bacteria is that they came in two quote-unquote flavors. If you plated these bacteria on a bacterial culture plate, they could form either rough colonies or smooth colonies. I can show them here. So there were the rough colonies and the smooth colonies. And he also knew that these bacteria were very different in a peculiar way. If he injected the rough colonies into a mouse, the mouse, a few days later, weeks later, was totally fine, living and breathing and walking around and doing mouse stuff. But if he injected the smooth bacteria or bacteria from the smooth colonies into the mouse, within days, the mouse would be dead. And he could take blood from those dead mice, plate that blood on bacterial culture plates, and the smooth bacteria would show up again. Do I have a... I don't think I have a slide for that. But anyway, so the smooth bacteria killed the mouse, the rough bacteria didn't. So my question to you, and some of you may have read already some of the material for this week, so you may kind of know the answer, but how could these S bacteria be killing those mice? So what could those S bacteria be doing that the mouse would be dead? So think about it for a second, talk to your neighbors, and let's discuss some answers. What was killing those mice? Yeah. Pneumonia, that's the disease, right? So how were those S bacteria causing that pneumonia? What were they doing to kill the mouse?
Maybe the DNA structure is slightly different that could disrupt the process in the body, or it then cause the pneumonia.
So the DNA structure, right? You went a few steps ahead, but there was something about those bacteria that was causing the pneumonia. Okay, excellent. So if I may paraphrase, those bacteria are producing some sort of venomous or poisonous or toxic substance. So one possibility was toxins, right? Let's go with toxins. Any other ideas? how else could these bacteria injected into a mouse be killing the mouse? Yes?
Somehow it was modifying its genes.
Whose genes?
The mouse's genes.
The mouse's genes.
Bacteria was modifying the mouse's genes.
Okay. Can we go with that is a different version of the same idea that the bacteria is producing something that is very somehow toxic to the mouse, in this case to its genes? Yeah? So along the same lines. But how else could these bacteria be killing the mouse?
A stronger defense against the immune system?
Stronger defense
Yeah.
against the immune system. So the bacteria defending themselves against the immune system. Okay. And if the bacteria are defending themselves against, or let me turn that around. If the immune system is really bad for the bacterium, how could it be bad for the bacterium? It eats it, right? So now these bacteria are not, if I'm understanding correctly, are not getting eaten by the immune system. So what can they do now? Multiply. There you go. So that was the other possibility, that these bacteria were not necessarily producing any nasty toxins, it's just that they're able to reproduce inside of that mouse, and not much different from, say, a cancer. They take over the organism, right? So they grow inside of the mouse, and it's like one of those zombie-like stuff that grows inside of you and ends up killing you, right? So it could be toxins, or it could be by division. Excellent, excellent. And that's exactly what was going through Griffith and whoever was working with him at the time, these different possibilities, right? So how could you test that idea? Like, let's go with your idea that it's actually they're just dividing. They're not necessarily producing a toxin, they're dividing. What could you do to test your hypothesis? And the question goes for everybody, it's not only for you. But let's say that the idea now here is those bacteria are getting injected, somehow they evade the immune system, and they continue dividing, they kill the host. Versus those bacteria were producing something that is so poisonous that they're killing the mouse. So how could you distinguish between the two?
Antibiotics.
Oh, I'm going to hold on to that key term, antibiotic. So what do you do with an antibiotic?
Eliminate bacteria.
Eliminate bacteria, kill the bacterium. So we're going to play off of that. So yeah, one possibility was to kill the bacterium before you inject it. If the bacterium was producing some sort of poison and you're injecting dead bacteria full of poison, the mouse should die anyway. Whereas on the other hand, if you injected dead bacteria that cannot divide anymore, then they shouldn't kill the mouse. Everybody follow? So that's exactly what Griffith does. He takes the bacteria, the S bacteria, kills them outside of the mouse by boiling them essentially, and injects them into the mouse. There's an assumption, hidden assumption here, that by boiling the bacteria, you're not destroying any poison they may produce, but it's worth a shot, right? So the idea here is I kill the bacterium outside of the mouse, I inject it. If it carries with it some sort of poison or toxin, the mouse should die anyway. But if it doesn't, then there was something about those bacteria having to be alive. And that's exactly what he saw, the latter, right? So the heat-treated S bacteria did not kill the mouse anymore. The mouse was alive and kicking weeks later. Okay? So it isn't anything that the bacteria was producing. It was something about the bacterium just being alive. Now we know a little better. We know that, and it goes perfectly with your thinking, with your hypothesis, that what these S bacteria are producing is a thicker coat around them so they can evade the immune system. So they're not producing anything nasty inside of the mouse. It's just that they can divide more without being attacked and eaten by the immune system. All right, so the next step was to say, all right, well, is there anything in this bacterium that could be transferred to a different bacterium that we could see as the carrier of genetic information? Normally, in genetics, people were concerned with different generations, right? What progenitors might be transferring to their progeny. What Griffith thinks is there anything that I could make these dead bacteria transfer to a live bacteria, and that would carry some sort of genetic information. So the experiment he tries is this. He kills the S bacteria with heat again, so he boils the bacteria, and now he mixes up the dead S bacteria, the boiled bacteria, with some live R bacteria. And if you guys remember, the R bacteria were harmless to the mouse. But if he mixes them up prior injection to the mouse, into the mouse for a little while, and then he injects the mix inside of these mice, now the mice die, and like when he was injecting the S bacteria alone, if he plates a little bit of the blood from those dead mice, he sees S bacteria growing in those plates again.
So I just want to make sure I'm understanding. So basically, he killed the S bacteria and caused cell lysis, which then he added R.
Let's say for now, he probably didn't know exactly what was going on. By boiling the bacteria, I think it's fair. People knew that by boiling any living organism, you would probably break it into bits. That's fair to assume. So whether or not he thought in terms of lysis, I don't know. But the idea here was that he was mixing dead bacteria with live, harmless bacteria and somehow making them harmful, right? And in fact, he proposed that there was some sort of transforming factor. There must have been some factor that was transforming the harmless R bacteria into harmful, lethal S bacteria. So there was something in those dead bacteria that was being transferred to the R bacteria and transforming them into R bacteria. Now, that was not the only interpretation of these results, and I'm inviting everybody to come up with an alternative explanation. What I just told you is what he proposed, right? But faced with the same data, can you think of any other explanations for the results? And mind you, the experiment was to mix dead S bacteria with live R bacteria, injecting the mix into a mouse, and seeing that a few days later to a couple weeks later, the mouse died and it was full of S bacteria. So what alternative explanation to this idea of the transformation can you think of? Excellent, yes! And you started saying, I don't think that's what happened. And now we know it's not what happened. But it's a totally logical possibility. And that's what these guys had to deal with back then. And that's what many of you hopefully will have to deal with in a life of research if you choose to pursue one. That results are not always so clear-cut that they have only one interpretation to them. They may have alternative interpretations. So what you need to do is design experiments that allow you to pursue each of those. I got so excited that I sidetracked the idea. But the idea is what, your name? Evan is saying, maybe somehow, I don't think this is what happened. I think this is what, but maybe somehow the live R revived the S. And that was totally consistent with the observations. If the S had somehow been revived by some bacterial CPR, now there will be some live S injected into the mouse, and it will do what S bacteria do when injected in a mouse and kill the mouse, right? So how do we test that idea? Well, that came years later. A group of investigators, Avery, McLeod, and McCarty, decided to say, what they said is, well, just to make sure that we're not putting into the mouse dead S that may have been revived through mini bacterial CPR. I'm going to overuse that joke because I think it's funny. So until I hear some laughter, we're going to keep hearing. So to make sure that we don't run the risk of injecting revived S, what we'll do is we'll take those dead S bacteria, split them up into their components, right? The sugars, the DNA or nucleic acids, the proteins, and the lipids. Use each of those separately, mix them up with harmless R bacteria, and see if any of these could be that famous transforming principle of Griffith. So by doing this experiment, these guys were killing two birds with one stone. On the one hand, they were addressing more directly what that transforming principle could be. So they're going like, well, we'll go with Griffith, and we'll assume that there is such transforming principle. So we'll take a step further, and we'll try and figure out which of these molecular types that transforming principle might be. And also, we're addressing this issue of, could it be revived S bacteria instead of transformed R? Okay? So the experiment they did is they heat treated, or they killed by boiling the S bacteria as usual, and then they used biochemical techniques to separate from that soup of the bacteria the nucleic acids, the proteins, the lipids, and the fats, right? And what they, particularly what, this is going a little deep into the story, they never tell us if they did the experiments with the lipids and the sugars. Probably they didn't, because biochemists knew enough about DNA and proteins that they knew there was enough variability in their structure and their chemical composition that it made them good candidates for carrying information, whereas sugars and lipids, I don't know how much you guys remember from your intro bio classes, but sugars are usually glucose, so there isn't really much room for carrying information. Yeah?
So you said, like, hypothetically, let's say that the R kind of flipped the S and made it...
Revived it.
Again, revived
Yeah.
it. So instead of splitting up the S's, why didn't they just try putting the S, like the live S bacteria with the R, and then like see if that could have like flipped it and maybe killed the...
Well, so the other way around, but
Yeah.
then you're knowingly injecting live S, and that was going to kill the mouse from the get-go, regardless. Yep. So the idea of can bacteria revive one another when one is alive and the other one is dead? Can you see that happening? The problem is that with this experimental setup, they would not have been able to tell if what's killing the mouse is a revive, and in fact, it would not have. The mouse wasn't going to survive anyway, even if some R had been revived. I appreciate the question, but it was a limitation of the experimental setup. But good question. Yeah.
Couldn't instead you could have just had a petri dish of R and S and see which one would have been more dominant, I guess? Or because if you would thought that the S made a toxin, you could see if the R survived it or not.
The R survived the toxin of the S.
Yeah, to see like if you thought that there was a toxin.
Yeah, but you have to understand that it isn't so much a toxin from the S that would kill the R. It was a toxin of the X that would kill a mouse. So it isn't so much a battle between them, it's what they can do to the mouse.
Can they do like an in vitro experiment where there's like in a glass they just see what happens to the S and R bacteria if you lysate the...
Yeah, so that if they could see some sort of in vitro revival of the bacteria. The idea is excellent, and I honestly don't know if they did it or not, right? What we know from textbooks is this experiment, because these address more what the transforming principle was. But I totally agree. They could have, in principle, tried some sort of bacterial revival in vitro. The problem is that, let's say you mix S, dead S, and live R, and now you see some live S. Was it a revival of the S or transforming of the R? And in vitro, that's impossible to distinguish, okay? But good idea, too. So the experiment that Avery, McLeod, and McCarty did was to separate the proteins, mix them with harmless R, and in a separate tube, take the nucleic acids and mix them with harmless R, let them incubate for a little while, and then they injected the mix into a mouse. When they had mixed the harmless R with proteins from S, from dead S, the mouse was still fine. So proteins could not be the transforming factor, right, or transforming principle. But when they mixed harmless R with DNA or nucleic acids from the dead S, now the mouse is dead, and then they can separate or they can isolate from that dead mouse some S bacteria. So this indicated that seems like it's nucleic acids that are the transforming principle of Griffiths. Now, they were not still totally convinced. I was going to invite some conversation, but I'm afraid that we're going to run out of time. I'm going to tell you why. So there was an alternative explanation for these results, and that had to do with what these people knew about biochemistry in those days. They knew that every time that you use biochemical methods to separate proteins and nucleic acids, there is more often than not some cross-contamination. What that means is no matter how hard you try, when you're using biochemical methods to separate nucleic acids from a sample, there's always going to be some proteins that are present in your sample and vice versa. They knew that. And they couldn't really avoid it because the biochemistry of these things are similar enough that it's just too hard. So at some point, someone within the group or some competitor or something must have said something along the lines of, well, but how do you know that your DNA is really pure? Maybe there is some protein there, and that is what's carrying the genetic information, right? Or the heritable information. So they decided to do a complementary experiment, and that is, instead of adding the DNA from the dead S to the R, what they said is, let's take dead S and let's take out each of these types of biomolecules one at a time. So instead of adding a biomolecule, they decided to take it out from the original mix. So what they did is they heat-treated S, and they treated those boiled bacteria with enzymes that would chew up either nucleic acids or chew up proteins. And just when you're thinking, well, but like, how do they know about these enzymes? They knew about these enzymes back then because many biochemists spend their days isolating different proteins from different biological sources and trying to see what they do. So for instance, in the early 1900s, people already knew that tears, human tears, are full of enzymes that will chew up proteins. And now we know that's a defense mechanism. When, of course, we cry when we get sad, but we also cry as a defense mechanism when we have an allergy or when we have the flu or whatever, to protect against the entry of any pathogens, right? So we produce all of these proteases that will go destroy anything that has proteins in it. Anyways, that was a slight detour. But the thing is that they treated the soup of dead S with DNases or proteases, and when they treated with proteases, meaning that they killed all of the proteins, the S bacteria could still transform the R, right? So taking away the proteins didn't really do much, but when they treated those dead S with the DNAs, now the mouse survived. So by taking away DNA, now those dead S couldn't transform the R anymore. Okay? Did everybody follow? What I wanted to do is take a very brief detour here to address how these experiments illustrate so nicely a principle that to this day is very dominant in genetics. And that is the principle or the concept of how you address experimentally, how you test experimentally any hypothesis of causality in genetics, right? So if we say in general, cause C leads to outcome O, I'm asking everybody now, how can you test that? If you have the following hypothesis, cause C leads to outcome O, how can you test it?
Do C and CFO happens?
Say again?
Do C and CFO happens?
So basically, do they occur together all the time, right? Let's say that they do occur together all the time. What's that famous saying that people love to throw around? Correlation doesn't mean causation, right? So we need to go a little further than that. So C causes O. How can I experimentally test that? Remove C, right? Remove C. Oh, I had add C first anyway. But yeah, remove C is the other one. So we can either add C or, let me see, there you go, or take C out, right? So if you're thinking C causes O, you have these two major avenues. One is you take a system without C, you add it, and now you ask the question, do I see O now? The other one is you take a system that already has C, take it out, and you ask, is O still there? These are, in very abstract terms, two types of experiments that are very common in genetics. When you add something to a system that didn't have it or had it in very low levels, you're doing a gain of function experiment, because now your system is gaining a function that it didn't have before, right? And we say that those are tests of sufficiency, essentially because what we're asking here is adding C sufficient to CO? Right? When we have a system that already has the cause and you somehow experimentally take it out, we say that those are loss of function experiments. Your system is losing a function that it already had. And what you're doing there is a test of necessity, right? Or requirement. You'll see those two terms thrown around quite a bit. Essentially, what you're asking is C necessary for O? Okay? Now, I said that I wanted to take this detour from the Avery-McLeod-McCarty experiment because that's exactly what they did. They were trying to link DNA as the cause of the transformation of the R. So they were trying to establish that causal link between DNA and transformation. So what they did is, in one experiment, they took DNA and added it to the R bacteria that presumably didn't have it. They did have DNA, but not that one, right? So they were doing a gain of function experiment. And they were showing that DNA is sufficient. Let me write that down. DNA was sufficient for transformation. In the other experiment, in which they took the whole soup of dead S and treated that soup with DNAs, they're taking DNA out. So they're doing a loss of function experiment. And what they were showing there is that DNA is necessary, or like I said, required for transformation. Now, as I mentioned briefly before, there was still a persisting concern that pure DNA wasn't ever really pure, because these molecules are biochemically very similar, and when you extract them from complex living organisms, there is a chance that there'll always be some degree of cross-contamination. So, some years later, almost 15 years later, Hershey and Chase at Caltech, so like people like to say up the road, they did this experiment. They took bacteriophages. Bacteriophages are a family of viruses that won't infect us. They will infect bacteria. Okay? And these viruses are not totally unique, right? Because human viruses can be the same as well. They have this feature that they don't have sugars and lipids. They're just nucleic acids, in this case DNA, encaged in a proteic cage, right? The cage we call the capsid, and the nucleic acid that they have inside is DNA. Those are the two components of a virus. Any other living organism that you think, or any living organism that you can think of, will have the sugars, the lipids, and all kinds of other types of molecules. These bacteriophages, proteins, and DNA. So they knew that these bacteriophages could kill bacteria. Switching gears here, we're not using bacteria that kill a mouse anymore. We're using bacteriophages that kill bacteria. And they knew that they're made of protein and DNA only. Now the question became, what is that the bacteriophage is introducing into this bacteria to kill it? And the reason I say this is microscopy at the time was advanced enough that there were many very good pictures of bacteria with these capsids all over its surface. So the capsid is on the surface of the bacterium, never goes in, and yet the bacterium dies. So the thinking was, these viruses, these bacteriophages, must be putting something into the bacterium. But what wasn't clear was whether that something that they're introducing into the bacterium is DNA or protein, or maybe both. So the experiment they did is as follows. They took these viruses and produced them in large scales in either a culture medium that had radioactive phosphorus or in a separate tube, a culture media that had radioactive sulfur. And the reason they did that is because they knew that that phosphorus as an element will only be present in DNA, not in proteins. Proteins, amino acids don't have phosphorus in them, whereas DNA is full of phosphorus. If you guys remember your DNA structure, it's the phosphodiester backbone, so every 2 nucleotides there is a phosphorus atom there. So if they grew these viruses, if they made these viruses in a medium that contained radioactive phosphorus, those bacteriophages would have radioactive DNA. In the other culture medium with radioactive sulfur, sulfur can go into certain amino acids and therefore proteins, cysteine being one of them, right? So with growing the bacteriophages in a culture medium with radioactive sulfur, they would create bacteriophages that had radioactive proteins. So now they have these two collections, right? They have the bacteriophages with radioactive DNA and the bacteriophages with radioactive proteins. And the question became relatively simple. They said, let's let these bacteriophages do their thing of coating a bacterial cell on the surface, inject whatever they need to inject, and then we'll look inside of the bacterium if there is any radioactivity or not. And what they found is that when the bacteriophages had radioactive DNA, they could see a lot of radioactivity inside of the bacterium. But when the bacteriophages had radioactive proteins, the bacteria were empty, no radioactivity in them. And yet they were dying. So what this experiment demonstrated is that these bacteriophages could inject DNA only, kill the bacterium. They didn't have to inject any protein. They could still be lethal to those bacteria. Now, how is this much different from the experiments from Avery, McLeod, and McCarty? It all comes down to sensitivity. Because no matter how hard you try to separate these things from living systems using biochemistry, this thing was much more sensitive because with radioactivity, you could detect the tiniest amounts. So the fact that they couldn't see any radioactivity when the bacteriophages had been grown in radioactive sulfur indicated that no protein goes into those bacteria. The issue that Avery, McLeod, and McCarty faced was, well, but you don't know, maybe you have a minuscule amount of protein and that's carrying information. Now these guys knew that there was no protein going in because they would have detected it. Radioactivity, the detection of radioactivity is sensitive enough that they would have seen it. Any questions? So this was an experiment that illustrates something that is, in a way, this was confirmation of ideas that have been thrown around for a while now. In fact, by the time that Hershey and Chase came out with this experiment, it was one of those that people say, well, that's the nail in the coffin, the proverbial coffin of the dispute, whether it's DNA or proteins, because now we're sure. But by now, people had been so convinced that DNA was the carrier of genetic information that they didn't really need to wait for this type of super elegant confirmation, right? So this experiment is historical, and we teach it, and we discuss why it was so important in the history of molecular genetics for how elegant it is, right? But the reality is that by the time that they came out with this experiment, everybody was pretty convinced that DNA was the carrier of genetic information. Any questions? I'm going to pause here for a sec, make sure that, yeah.
So the black virus is the phosphorus and it's injecting sulfur into
No. So the radioactive phosphorus would be here, this yellow string.
okay, got it.
Yeah. So in this cartoon, yellow is radioactive, black is non-radioactive.
So what's being injected in though is the DNA strand, right?
Yeah, so what this cartoonish again, but what they're injecting is the DNA strand, and once inside of the bacterium, that DNA will be, as we'll see next week, transcribed and translated into what? into a whole bunch of viral proteins that will kill the bacterium from the inside. And replicate it. I missed that, but this DNA will be replicated into many, many, many copies of new viral DNA that will be packaged into new capsids, make new viruses that will go attack and infect the next bacteria. Any more questions, anyone? No, we're good? Okay, let's move along. So, almost last page. So in 1953, some of you may have heard this story, Watson and Crick published their double helix model for the structure of DNA. So to place you historically, in 1952, Hershey and Chase published their paper with it's DNA. But people had been already after DNA as the genetic material for a while because they had been convinced enough by Avery, McLeod, and McCarty. Watson and Crick were among them. And now the question became, okay, we know biochemically the composition of nucleic acids. It's an acid. We find it in nuclei. We call it nucleic acid. We know that it has sort of these biochemical composition, but we have no idea what it looks like in real life, what its structure is. And everybody is desperate to find the structure of DNA with the hope that knowing the structure of DNA, they would figure out how it worked. So what is that Watson and Crick knew? They knew that DNA is made of these 4 building blocks, these 4 nucleotides that we call, for short, A, T, G, and C. You guys obviously heard about this before and probably studied in some detail. They knew their chemical structures because people had broken up DNA into these building blocks and, again, using organic chemistry this time, they had figured out what these things looked like. To the point that, story has it, these guys had cardboard cutouts of these molecules. Today, we would use those string and ball models that you would see, in high school and whatnot to make little atoms and molecules and elements in 3D. They had cardboard cutouts of these things, and they would use them to figure out how they could try and fit them together to try and understand how these molecules interacted with one another in a DNA molecule, okay? So they knew the chemical structures of A, T, G, and C, and they also knew, Chargaff was a scientist who had, a few years before, come up with this sort of rule. They called it Chargaff's rule. Any time that anyone isolated DNA from any living organism, when they went through the organic chemistry or biochemistry steps to separate these A, C's, T's, and G's from one another, they would find that at the end of the day, the amount of A in that DNA was always the same as T, and the amount of G was always the same as C. Okay? So no one had any idea of what that meant, but it's something that was observed over and over and over and over. So they have these things. And again, story has it that one day Watson takes a trip to visit a colleague/competitor named Rosalind Franklin in a university nearby. And when they're chatting, Rosalind Franklin, not even her, but an assistant of hers, shows Watson a famous picture that they had taken of DNA crystals. Okay? I've been doing this for almost 30 years. I still wouldn't know how to interpret those pictures. I'll be honest with you. Those are X-ray diffraction, crystallography stuff. I don't know how to read any of this, but Watson knew. And he saw those pictures and he's like, oh my God, this says to me that DNA must be some sort of double helix. So he goes back to the lab, and now knowing that it is some sort of double helix, it now became a lot easier to put these cardboard pieces together and make them fit into a model that would be consistent with all of the experimental observations that people had done at the time. Almost like when you're starting a puzzle, and when you guys start a puzzle, one of those large ones, the 1,000 pieces puzzle, what is that most people do first? The edges, right? Same thing. So this thing gave them a starting point to start playing around in a much more constrained space of possibilities. Now they knew that there had to be some sort of double helix, and it wasn't long until they figured out their famous. There you go. Thank you. You didn't want to interrupt the show, of course. Thank you. This is such a 21st century problem to have. So they came out with their famous double helix, where T's base pair with A's and C's base pair with G's, explaining the famous Chargaff rule. So this was sort of the chemical basis for why there was always same amount of T's as A's and same amount of C's as G's. It was simply because that's how they paired with each other between the two strands of the double helix. They figured out the anti-parallel orientation of these strands. We're going to revisit some of these later in the semester, but some of these may sound familiar to you. And they came up with the double helix structure. And it was revolutionary. Now everybody not only knows that DNA is the carrier of genetic information, but also what it looks like, right? And that triggered a whole bunch of new lines of investigation related to how these double helix would contain genetic information, express that genetic information, right? So how do we go from these double helix to the proteins that are, at the end of the day, doing the work, right? Because that's another thing that people knew. It's one thing to carry genetic information, but how do living organisms function? That was mostly through proteins. So somehow we had to go from information to action, right? But all of that became a lot easier when you knew where you were starting from. On that note, I'm going to invite everybody to not forget to check out a couple of videos. It's essentially sort of a recording of everything that we just discussed. And in particular, this one that discusses the Meselson and Stahl classic experiment that addresses how these people discover how DNA is replicated. Okay, so let's finish this brief recount of the history of molecular genetics with the famous Beadle and Tatum experiment. So everybody knows DNA carries genetic information. Now, like I said before, the question became, how do we go from that information to action, right? So how do we go from the DNA that carries the information to the proteins that we know do the stuff, okay? So they decide to address a question that was everywhere at the moment in the mid-40s. And that is, notice that it's before the structure of the double helix or Hershey-Chase's experiment, right? But even in the early 40s or mid-40s, people were so convinced that DNA was the carrier of genetic information anyway that they went ahead and like, oh, we're going to start investigating the next big question, and that is, how do we go from information to action, right? So one of the questions that was lingering is, how is information coded in that DNA? Is it globally coded or is it fragmented? So people knew that DNA was a very large molecule, and they also knew, obviously, that living organisms passed a lot of information about various things to their progeny. So the question became, is all of that information encoded globally throughout the entire DNA molecule, or is it fragmented such that little pieces here and there contain information? So they decided to address that question using a fungus. And now the fungus that immediately may come to mind, the mushroom body and the, this is a unicellular fungus that grows as a film on culture media. And if you let it grow in bread, it'll be mold, essentially. But in the lab, it grows as a film. And what they knew about this fungus is that they can grow in media where you give them everything they need, vitamins, amino acids, sugars, everything. But they could also grow in minimal media with only sugars and salts. What that means is that these fungi, like many bacteria, they can make everything from scratch. Everything they need, they can make from scratch. We can. That's why we have bacteria in our guts, because they give us the vitamins and amino acids that we can produce ourselves, blah, blah, blah. But this fungus was totally autonomous. If you give it salts and sugars, it can make everything else from scratch, right? So they knew that. They also knew that if you irradiated these fungi with X-rays, they could induce localized damage in DNA. And how do they know that it was localized? Because when they irradiated these fungi with X-rays, it's not like the entire DNA in these fungi vaporized and was completely lost. No, it just came out in shorter fragments, indicating that it was just being broken into small pieces at localized positions. So they know that these fungi have all the information they need to make everything from scratch, and they know that DNA can be damaged at localized positions with X-rays. So they decide to put the two pieces of knowledge together into asking this question. If we can damage DNA locally, will we affect the ability of these fungi to make specific amino acids and vitamins, or are we killing their ability to make them all? Any computer science majors in the room by any chance? So I can say whatever I want about computer science, you won't be able to. I once heard, I don't know, that computers, when you store something, when you store a file in a computer, is not saved in a hard disk in a specific place. So your Word document doesn't go into one place in the hard disk. It actually gets fragmented and saved into little pieces here and there. I don't know if that is true or not. I heard that once. But that was the thinking back then. Maybe the information to make certain amino acids is distributed throughout the entire DNA molecule, and anywhere where I cause damage, I will damage that information. So I decided to test this, and the way they go about this is they take these fungus, Neurospora crassa, and they decided to mutate it with DNA and basically ask the question, how many abilities did they lose? If information was distributed globally, no matter where that damage was, many abilities would be lost. But if information was localized and fragmented, then some abilities would be lost, not all of them, or not many of them. Does that make sense? If you didn't get it right now, spend some time with this slide later, because that's the essence of the experiment. The rest is bells and whistles. We'll go through them, but this is the sort of the essence of the experiment. Understanding that if information is globally coded, damage anywhere in that DNA would break a lot of information. Whereas if information is localized and coded, restricted to specific location, if you miss that location with your X-rays, that information is fine. Okay? And because X-rays could cause limited damage here and there, presumably you would miss most of the information needed by these fungi. All right, so how do they go about this? They mutate the fungus. They have to shine X-rays into a large collection of fungi. Now, this mold grows as an entity. You can see it as this film in the tubes with the culture media, but in reality, it's a whole bunch of individual fungi cells, right? So it's a whole bunch of individuals. So you shine them all, and you know that you're going to be causing localized damage in different places in each of those fungi. So then they could, using fine needles, they could pick up individual spores of those damaged fungi and seed them individually into rich media. And they asked the question, which of these fungi can grow again into a new film? That was important, that after damage, these fungi would be able to grow again. And the reason it was important, why was it important? The reason it was important is because when they irradiated these fungi with X-rays, what they were trying to get at is this connection between localized damage and the ability of these fungi to make certain molecules, certain amino acids, certain vitamins. But making certain amino acids and certain molecules is not the only thing that a fungus needs to grow. It needs other things. It needs the ability to replicate DNA. It needs the ability to make new cell walls. It needs the ability to divide mitochondria. It needs the ability to A, B, C, D, and E, right? So they needed to make sure that whatever damage they had caused to this fungi had not killed their ability to grow. So they would damage the fungi and then seed them again in rich media. If a fungus could not grow in rich media, it meant that something else was broken. Right? Cool. So now they have a collection of fungi that, even though damaged, could grow in rich media. So they hadn't really lost that ability to grow when given everything they needed. The next step was to see what would happen. So this was to determine that a strain could grow when given everything. The next thing was to determine if they could grow when they were not given everything. Right? So they would take, now that they have these different mutants, mutant X, mutant Y, mutant Z, mutant A, B, C, and D, they would individually seed them again into rich media or minimum media. Some of those mutants could grow in the rich medium, and they could also grow in the minimum medium. So we don't know, they didn't know where the damage was, but what they did know is that that damage hadn't inhibited their ability to make all amino acids and vitamins. So immediately they were not interested in those mutant strains anymore. The ones that caught their eye is these strains that would grow in rich media, so they grow when you give them everything, but they stop growing when you don't. Because that meant that those strains were missing something. Another question was, is this something they're missing, 1 or 2 amino acids and vitamins? Or are they missing the ability to make many of them? Because that went back or tied back to this question of, will localized damage affect one ability or will affect many? So what they did is they focused in on these strains that all of a sudden couldn't grow in minimum media. And what they did is super clever. They took that minimum media and started adding the amino acids and vitamins one at a time. Right? So what they would see is that in minimum media, those strains didn't grow. We knew that already. Minimum media plus alanine, they still didn't grow. So it's not that they needed alanine. They didn't grow in minimum media with leucine. It's not that they needed leucine. I mean, they needed it, but when you gave it to them, it just didn't change things. All the way down, and boom, let's say this is an example, but in minimum media with arginine, now all of a sudden the fungus grows again. So what this experiment was indicating was that these mutant strain, remember they had hundreds of them, this particular mutant strain had lost its ability to make arginine. That's it. That's all they were missing. Because when you put it back, they didn't need anything else. They could make everything else from scratch, but arginine. So all they needed to do was replace that ability externally by putting arginine in the culture medium. So now this mutant Y became a mutant that can't make arginine, and they would, and I'm not joking, they would name these mutants, let's say ARG1, LEU3, IS6, for the amino acid that they were missing and the number of the strain that was mutant for making that amino acid. So they went from not knowing absolutely anything to having a collection of mutant strains that couldn't make specific amino acids. Now, one thing they knew from biochemistry is that arginine is not made from sugars and salts in one step. Biochemists knew that to go from glucose and some sugars to making arginine, there are several chemical reactions involved, right? And if we think, it's very abstract, cartoonish, but conceptually, that you start from some substrate 0, and you go through a series of intermediates, substrate 2, substrate 3, 4, 5, 6, till you get to arginine, each of those chemical reactions are normally catalyzed in living systems by enzymes. So you go from substrate 0 to 1 through the activity of enzyme 1. And you go from substrate 1 to substrate 2 through the activity of enzyme 2, and so forth. So they could extend the same reasoning to figuring out which of these enzymes was missing in the biochemical pathway that leads to arginine. So what I mean by that is they would take ARG1 and then transfer a spore into minimal media that had substrate 0 or substrate 1 or substrate 2 and so forth. And they would notice, for instance, that when you added substrates 5 or 6, now all of a sudden the fungus could grow again. So what that was telling them is that, yes, they couldn't make arginine, but why? Because they couldn't make substrate 5. The second you gave them substrate 5 or 6, they could take it from there. They could take it from there. Or arginine, of course, the final product. Right? So this is how they would connect, for instance, that arginine 1 wasn't a mutant that couldn't make arginine. It was actually a mutant that couldn't make E5, the enzyme that takes substrate 4, turns it into 5. That's how localized, how restricted was the loss of information. So localized damage to DNA was damaging this one enzyme. And that's how they became with this idea that one gene means one enzyme. And this idea, this concept carried us all for a while. Now we know a little better than that. Now we know that sometimes it is several genes that mean one enzyme. Sometimes it is several genes that mean several enzymes, all of which contribute to an ability, right? But this was a huge step because essentially basically said DNA contains information in little packets, each of which has all the information that the cell or the living organism needs to carry out a function. Okay? And this was huge. It meant that the information was contained physically in little fragments of DNA. So now we can connect DNA to proteins through this idea of one gene, one enzyme. And I know I'm a minute over, but I'm going to leave you with this because there is a homework assignment in Canvas dealing with this, the central dogma of molecular biology. Now we know that information in DNA gets written into RNA molecules in a process that we call, anyone, transcription. And then the information in RNA molecules gets translated to to protein sequences. Now, the homework assignment has to do with how this depiction of the central dogma is a little incomplete. So make sure that you check it out. It's due sometime over the weekend, I think. And then we'll take it from here next week.